Good morning, student. In this video, I shall discuss the fingerprinting, that is the DNA fingerprinting, and which is one of the important application of the genetic material in case of human genetic material. Now, what is this DNA fingerprinting and how it is different from the normal fingerprinting? Earlier, this fingerprinting was taken as the finger of the human being, and this fingerprint is considered, or till now it is considered to be one of the unique features of the individual. So by this we can easily identify the uniqueness of that particular individual. But nowadays, as the technology advanced, now this DNA or the genetic material or the genome of the individual is taken as the uniqueness of the individual, and by considering this uniqueness, now the individual are identified properly. So what is this DNA fingerprinting, and what is the basic principles behind it, and next one, how it is done, and what is its application. These are the three important aspects of this DNA fingerprinting. Now, first of all, I shall discuss the principles behind this DNA fingerprinting. Now, in case of human genome, as we have already studied in the Human Genome Project, there are around 3 billion base pairs are present in the human genome. And out of these 3 billion base pairs, normally 99.9 genome are identical in all respects. That means all the individuals, although they are morphologically dissimilar from one another, they are physiologically dissimilar from one another. So whatever differences is there in the individual, that is only due to 0.1% difference in their genome. So 0.1% difference in the genome makes the individual unique. Now, whether it is morphologically so much different, but this difference, just you observe the physical appearance of a Negro people, the physical uniqueness of the Mongolian people, physical uniqueness of other people of different regions. So this uniqueness only due to 0.1% difference in the genome of the individual. Now, as we have already discussed, the genome or the DNA material which are present in the human being, these are of two forms. First one, that is the expressible form, which normally forms the mRNA, and this mRNA forms the protein. So these are called the expressible genetic material or the expressible part of the DNA. But there are certain parts of the DNA which are more than 70%, and these are unexpressible parts. And these unexpressible parts, normally these are structural DNA which are present in the centromere of the chromosome. Normally in the centromere region of the chromosome, they are structural in nature. They just organize the chromosome in a definite manner, and these are also the regulatory gene, or they have some regulatory function. So whatever the case may be, but they are more in number or more in the sequence than the normal expressible DNA sequence. Now, these DNA sequence which are normally present in this form or unexpressible form, there is certain random variation is observed in every 200 base pair. So every 200 base pair, randomly some sort of variation is observed. But it is not possible to observe this variation in whole entire genome of the individual. Suppose individual 1, there are around 3 billion base pairs, and there is variation in every 200 base pairs. But of course variation is there. We can observe it, but it is not physically possible. So that is why some methods which are developed to identify the uniqueness of the individual through this genetic material of the genome, that is the repetitive DNA. So in the part of DNA, some segments are repeated, and these parts are called the satellite DNA. And the satellite DNA on the basis of the length of the base pair, and these base pair, they are called either mini satellites when the base pairs are 10 to 60 base pairs in long, and it is normally G series. AT and GC, two types of base pairs are there. So it is G series, then it is mini satellite, and sometimes when the length is very short, only 2 to 6 base pairs, then it is called microsatellite. So in the DNA sequence or in the genome, there are some microsatellite, some mini satellite, and these are the variable region, and this high degree of variation in the genome. Then why this variation occurs? This variation occurs due to the mutation or change in the base sequence or the number of bases, and this variation may be somatic. It occurs in the somatic cell, and it occurs in the germ cell. Somatic chromosome and the germ chromosome. What do you mean by somatic chromosome? In case of human being, chromosome number 1 to 22, all are the somatic chromosomes. These chromosomes responsible for the expression of the somatic character. But the X and Y chromosome, these are called the germinal chromosomes or germ chromosomes or allosomes, and they are responsible for the reproductive character. So that is why there is variation in these regions, either in the somatic region or in the germ chromosome region, and these variations or these variables are normally used as the genetic marker for the uniqueness for that particular individual. Now, what is this genetic marker? So it is the variation in exon region. Exon and intron regions are there in case of mRNA. So there are some exon regions which are normally do not code for any protein. So these sequences are normally used as the genetic marker for the individual. Now, these genetic markers are of two types. One is single nucleotide polymer. Now, what is a single nucleotide polymer? Look at the example here. This is the example of a single nucleotide polymer. And here G, T, S, C, these are the base sequence. And in this case, you will find T. Suppose it is individual 1 and this is individual 2. And in individual 2, in this particular case, you will find G, T, A, C, and here A is present in place of T. In the first individual, there is T, and in the second individual, there is A. So that is why this difference in the nucleotide, this is the single nucleotide polymer, or it is called SNP. And this type of SNPs are observed in the nucleotide chain. So that is why it also used as the genetic marker for the identification of the individual. Now next one, short tandem repeats. Short tandem repeats, suppose these are the short tandem. Here ACT is taken as the example for short tandem repeats. Now the ACT, there are 5 repeats. Here in this case, 5 repeats are here. So the repeats are 5 in number. In the second case, the repeats are 6 in number. So that is why this 6 repeat and 5 repeat, this makes the individual unique from one another. So that is why these are the two important repeats which are normally used. First one, the single nucleotide polymer, and second one, the short tandem repeat. Now number of repeats, it normally vary from person to person, and the VNTR regions also, these are vary from person to person, and that is why it is taken as the genetic marker for its identification. And this technique was first time developed by or discovered by Alex J. Jeffrey, and the southern blotting technique was adopted for the entire process. That is why it is easier for this identification process. Now, next we shall discuss what is the procedure and what are the methods by which this DNA is normally isolated and they are properly identified. Now we shall discuss the technique behind this DNA fingerprinting. The first part of this technique, or the first step of this technique, that is the isolation of the DNA, because the DNA is present inside the nucleus of a cell and we have to isolate that DNA from the nucleus. And after that, the digestion of the digestion by the restriction endonuclease enzyme, because this endonuclease enzyme, it is the molecular scissor which normally cuts the DNA into a particular part of it. So that is why this enzyme is used as the digestion for this DNA long chain. Now this DNA long chain, which is now divided into number of fragments with the help of the endonuclease enzyme. Now these fragments are separated by the electrophoresis process, because there is a separation method, which is centrifugation, and normally they are separation by this electrophoresis method. The relative migration of these fragments on the basis of their molecular weight, normally they are separated. Now after that, after the separation, they are transferred them into the nylon sheet, and when it is transferred, normally after that they are taken the UV radiation photographs, and after that they are hybridized by using the labeled VNTR probe. The non-VNTR probe are used for this hybridization process. Then when they bind with them, automatically their sequence is easily identified. Now after the detection of this hybrid DNA fragment, normally this autoradiography technique is used, and by this autoradiography technique, normally these are clearly identified which segments are present there. Now from this, by comparing the probe and with the help of this technique, the DNA fragments which are isolated and on the basis of the different chromosomes, suppose this is the chromosome number 1, chromosome number 7, chromosome number 2, or chromosome number 16, whatever may be, then what are the different small tandem repeats are there, and you see these small tandem repeats are normally analyzed here, and after analysis, suppose this is the report of a crime scene. Suppose in a crime scene, it is found that some DNA are detected, or some, you can say, the some cells are normally recovered from that crime scene, and a single cell is enough to conduct this DNA fingerprinting process. So when this cell is found there, either it is a hair cell, or it is a blood cell, or it is a semen cell, then whatever may be the cell, we can recover the whole DNA genome of that individual. Now this genome is prepared, and that is called crime scene cell or crime scene DNA probe. And this crime scene DNA segment, it is now, suppose example, it is a chromosome number 7, and this is chromosome number 2, and this is chromosome number 16. Now there are some suspects. Suppose it is individual A and it is individual B. These are the two suspects, and we took the DNA of these two individuals. Then it is isolated, and after the preparing this DNA fingerprinting technique by adopting it, now when it is prepared and now it is compared. Suppose this is the crime scene cell, this is individual A and suspect individual 2. Now after the comparison, it is found that the individual B is the accused one, because this DNA fingerprinting of this one, this crime scene, it matches with this one. So that is why from this technique it can easily be identified that this individual is the criminal. Now what are the major application of this DNA fingerprinting? This DNA fingerprinting, normally applied in the forensic analysis, as we have already discussed. In the forensic analysis, normally, suppose in a crime scene, we found certain crime scene DNA material, or you can say some cells of an individual. Then this DNA fingerprinting is prepared. After that, the suspect individuals were normally detected, and from those individuals, the DNA fingerprinting is prepared. Then we compare, and after comparing, we can easily find out the particular criminal. So that is how the problems can be solved easily. Next, the paternity dispute or maternity cases. Who is the parent of that particular individual? Suppose a young one, there is dispute of paternity case, or sometimes there is dispute of maternity case. At that time, this DNA fingerprinting, which is cross-matched with the child and the child with their father or claimant of the mother, and when these were cross-matched, the paternity and maternity cases are resolved. Now, the anthropogenical studies, normally, suppose an individual, a race of human beings, they are originated from this one or originated from this one, whether they are migrated from this place or migrated from that place. So there is a similarity between the individuals of a closely related one. So that is why the anthropogenic studies are also now based on the DNA fingerprinting technique. Now, pedigree analysis. Pedigree analysis means how the hereditary diseases are the problem. This normally carried out from one generation to the another generation in a family. So this can also be analyzed, and this can also be studied properly with the help of this DNA fingerprinting. Now, the taxonomical studies, that means for the classification of the organisms into different groups. Earlier, where the morphological similarities were considered, then genetical considered. Nowadays, now this DNA genome or the DNA fingerprinting study is helpful for the taxonomic categorization or taxonomic classification of the organisms, to put them in a particular group, whether they are similar to one another or not. So that is why this is also helpful in this the conservation of the wildlife. There are so many wildlife in the nature, and for their conservation processes also this is used so that we can protect them from the nature. Now, this technique was developed by Alex Jeffrey, and the field is now extending, and so many other fields are also now taking the help of this DNA fingerprinting technique, and which will definitely be helpful for the human being. Now, I hope this video will be beneficial for you, and if really it is beneficial for you, then please like it and subscribe it. This completes your chapter, Molecular Basis of Inheritance. Thank you.